So I want to finish up our discussion on nucleic acids by talking about the information content that occurs in DNA. If we talk about RNA, we know that it has information content, we know that, but in addition it has multiple additional functions in the cell. Then we talked about gene expression and ribozyme activities and the possibilities of polymerization. So lots of activities of RNA, but DNA has one function and one function only as far as we're aware, and that is storage of genetic information. It has no activity on its own. Anything that is retrieved from it is retrieved either by proteins or by RNA. Now, in terms of the information content of DNA, most of the time we talk about the genetic code for protein synthesis. We'll talk a little bit about that because that's really what we know the most about. That was worked out in the 60s. Watson and Crick were involved with that. Also Sidney Brenner, who I mentioned when we talked about nematodes or C. elegans. And he didn't win a Nobel Prize for that, but he did win a Nobel Prize for his development of C. elegans as a model. But the simplest information that's contained within the DNA is the genetic code for protein synthesis. But I just want to say there's much more there. That is, there's much more information in the DNA. I'll just give you a simple example. It's not simple, but we'll talk about it later. Splicing, where introns need to be removed from an RNA molecule in order for it to become a functional messenger RNA. The information for the proper splicing is contained within the DNA sequence. In fact, if you have a protein that needs to go to the membrane, let's say, the information that it needs to go to the membrane is also present in the DNA. So there's tons of additional information. Whether a gene should get turned on or turned off, that information content is in the DNA. But the genetic code is what we understand the most, and that's of course what we have worked out for many years, and so we'll talk about that. So the function of DNA, storage of genetic information. And I just want to draw your attention to the genetic code for protein synthesis. And the first term that I want to introduce is the term codon. And each codon basically specifies an amino acid. And each codon is made of 3 nucleotides. And there's 64 possible codons. We'll talk a little bit about that in a few minutes. But if you look down at the line drawing at 5', 3' end, that represents actually that's a single-stranded DNA molecule. It's not RNA because it has T, not U. But the codons are made up of 3 nucleotides. So the first codon, ATG, would specify an amino acid. The next codon, CCA, would specify another amino acid, and so on. So on the right-hand side, you can see again how this would work. So CUC specifies leucine, AGC specifies serine, GUU specifies valine, and so on. So the gene contained in the DNA, the gene contains a long sequence where these individual codons then specify which protein is synthesized. So we refer to that as information. The DNA contains the information. Now, if we're looking at DNA sequences that encode proteins, then the ultimate goal is the protein. And the information is stored in the DNA. So that information is accessed in 2 steps. The first step, which I've written on the left-hand side, the first step is transcription. That is where you take the DNA, double-stranded DNA, and you synthesize from that an RNA molecule. Then that RNA molecule, which then carries that genetic information, is then brought to the ribosome, and that's where translation occurs, and then you finally get the protein. So this is illustrated kind of in a schematic form in the next slide. So this is what happens in eukaryotes, because eukaryotes have a nucleus, and that's in white. The cytoplasm is in gray. In bacteria, similar in a sense, but there's no nucleus. So here you have up at the top, you have double-stranded DNA. The nucleus, the introns and exons, we'll talk about later. But the bottom line is you get transcription. And when you get transcription, that refers to the synthesis of an RNA molecule from that DNA. And what it shows is after transcription, you get a pre-messenger RNA. Sometimes it's called a primary transcript. Then it has to go through a bunch of processing, which we'll talk about each one of these steps. The 5' cap, the splicing, polyadenylation. That takes place. But ultimately, what you get is a messenger RNA. And that's shown at the bottom in blue. And then that messenger RNA gets exported out of the nucleus into the cytoplasm to where the ribosomes are, and then you end up getting the protein translated. So the information flows from the nucleus out into the cytoplasm. And that's because the DNA remains in the nucleus. If the DNA were able to transfer out of the nucleus, and back in, maybe we wouldn't need the transcription part in order to make RNA. But the DNA remains in the nucleus. In order to get that information to the ribosome, where the protein is synthesized, we take that information that's encoded in the DNA, we make an exact copy of that. So the information is copied, just like if you put a thumb drive in a computer, you copy the information, and you move it into the cytoplasm where the ribosomes are. Now, if you look at the right-hand side, it shows at the top a double-stranded DNA molecule. It doesn't show the nucleotides, but it shows base pairing, and you can identify which is the G and which is the C, even with this, because the G's and C's have three base pairs, the A's and T's have 2 base pairs. And so you see the double-stranded DNA, anti-parallel, and what happens is, in order for transcription to occur, that DNA needs to unwind. Those strands need to come apart. The RNA polymerase then gets onto the DNA and then synthesizes, and what we're looking at here is it synthesizes the top strand. It puts down a nucleotide which pairs with the first one in green. And then it puts down the next nucleotide which base pairs with the nucleotide in yellow. So that's 5' to 3' synthesis, and it continues. And so it makes an exact copy. And that's referred to as transcription, and that's what you see in the blue in the middle. So that's the RNA molecule. Eventually it would end up being translated. Now, there's 2 terms I want you to know in addition to transcription, translation. There's what's called the coding strand and the template strand. Now, when the RNA is made, it's made using instructions on the template strand. So with green, you get red. That would be, guanine and then cytosine. Cytosine would be green, represents the larger nucleotide, which is guanine. And so you go down the line and you synthesize it. And if you look at the nucleotides, the red, blue, yellow, green, and so on, they're the same exact nucleotides as you have on the top. The only difference is there would be uracil instead of thymine, but we saw that they're kind of the same. So the sequence remains the same. And so the bottom strand is the template strand. That's what gives rise. That provides the template to synthesize the upper strand, which in this case is the coding strand. Why is it the coding strand? Because it encodes the protein. Why does it encode the protein? Because that's what's generated, that's what used to generate, no, let me say it this way. that is the same identical sequence as the messenger RNA, which we know codes protein. So the coding strand is shown here on the top. Doesn't have to be that way, but this just makes it easier. The coding strand's on the top, and the template strand's on the bottom, and it generates an RNA which has the coding strand. Now, if you look at the sequence below, it actually shows a nucleotide sequence, and you can see, CCC, ACA, and then GCCG, and so on. And that, in that case, is the coding strand, and the lower strand is the template strand. And when you look at the RNA molecule, you can see it starts at the beginning, C, 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 A, C, A. Up at the top, C, 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 A, C, A. And that molecule in blue, that RNA, will code for protein. So the coding strand contains the coding sequence for protein. The template strand is the complement to that, but that gives rise to the coding strand because of the specific base pairing. Now, when translation takes place, and we'll talk about this later in the semester, there are transfer RNAs which have what are called anticodons. That is, 3 nucleotides which base pair with the codon. So in this nucleotide here, in this sequence down here, if the first nucleotide is CCC, first codon is CCC, then what will happen is a transfer RNA with GGG will base pair to that. Then it will also be carrying with it the appropriate amino acid, which we'll see in a second is glycine. And so one by one, the codons get read by transfer RNAs that carry in the specific amino acids. And we'll look, when we talk about translation later in the semester, we'll look at the details of that. So the genetic code was worked out quite a long time ago. We understand it really well. That is, which codons encode which amino acids. And we've been able to generate a table that looks like this, where you can take any sequence and then figure out which amino acid would be synthesized. Or you can take any amino acid and figure out a codon which would give rise to that amino acid if you were to synthesize the nucleic acid, let's say. This is an RNA codon table, and we know it's RNA because there's U in red, uracil. If it was a DNA codon table, it would be T instead, but they're basically the same. Now, the way these things are read is you have the codon. Let's say the codon was AGU. AGU. So the first position is A, which brings you down into that third kind of section down below. The second codon is G. So you go up at the top, follow the G, bring that down. So it's either serine, arginine, or arginine. And if the last nucleotide of the codon is U, then it's serine. AGU is serine. AGG is arginine. If you looked at AUG, and I'm picking this for a particular reason, I'll tell you in a second. A is, again, that third quadrant. U is the first column. G is the last row in that column, last row in that line, and that's methionine. AUG is methionine. And the reason I'm pointing that out is because AUG is the start codon for protein synthesis, which we'll talk again about later, and that encodes methionine. So methionine is the first amino acid for every protein. Now I want to point out a few things on this table. If you have four nucleotides, and a codon is made of 3 nucleotides, it turns out there's 64 possible ways to generate 3 different codons using 4 nucleotides. But there's only 20 amino acids that are used in protein synthesis. So what happens is many of the codons, or many of the amino acids can be encoded by more than one codon. So if you look at leucine, for example, there's 4 codons. There's 6 codons that encode leucine. UUA, UUG, leucine. CUC, CUU, CUA, CUG, all encode leucine. So many amino acids can be encoded by more than one codon. There's a redundancy because there's 64 possibilities of these things and there's only 20 amino acids. And if you look at the table, you can see that most of these, all of them except for methionine, are encoded by multiple codons. Proline, threonine, alanine, valine, you see that they're all. So that's one thing, and that is referred to as degeneracy of the genetic code. Degeneracy of the genetic code represents the fact that multiple codons can be used to encode a single amino acid. The other thing I want to point out is, again, the methionine, which is AUG, the start codon. There's only one of those. And then I want to point out that there's 3 stop codons, UAA, UAG, and UGA. These are all stop codons. What that means is if they're encountered on a messenger RNA that's being translated, that's where protein synthesis stops. Okay, and when we look at protein synthesis and translation, we'll look at this again. But what I do want you to be able to do for an exam, let's say, is to be able to figure out, if I gave you a sequence of DNA or RNA, that you will be able to determine what the amino acid sequence is. So what we see here, what I'm getting at, is the information contained in the DNA moves into RNA by transcription, then moves into protein by translation. So there's a specific flow of the way the information goes, and that's from RNA to DNA to protein. Now that concept has been referred to as the central dogma of molecular biology. It's a specific information flow, and that's shown here. So the information flow refers to the central dogma of molecular biology. Originally, it started out as DNA to RNA to protein, and that is what happens. But it gets a little bit more complicated the more we learn. So you see many arrows here. The red arrows are the classic central dogma, that you have the red arrow going from DNA to DNA. Why is that? That's DNA replication. So the information is copied. DNA to RNA, that's transcription. And then RNA to protein, that's translation. So the information flows downwards. But we've learned that there are additional capabilities. So there is an enzyme called reverse transcriptase, which will copy RNA into DNA. So reverse transcription, RNA into DNA. And then we know that RNA replication can take place because there's RNA viruses and they replicate through RNA. So RNA to RNA through using an RNA polymerase, RNA-dependent RNA polymerase, that's RNA replication. So the information can flow in all different ways, but there is no arrow going from protein back to RNA. So RNA to protein is unidirectional. And if you think about it, it makes sense because DNA to RNA, DNA to DNA, RNA to RNA, or RNA to DNA is using the same material. It's all nucleotides. It's the same language, so to speak. But then the minute it goes into protein, it's a different language. It's a language of amino acids. And aside from doing it manually using a table, the cell does not have the capability of taking a protein sequence and then reversing that to synthesize, let's say, an RNA molecule. Okay, so that's basically what I wanted to talk about in terms of nucleic acids. We're going to move on, and we're going to start looking at chromosomes, what this DNA looks like in a chromosome.